Well, it's a pleasure to be part of the Academy and to give this presentation on the age of antioxidants. So that's my disclosure slide. So, atmospheric oxygen, 20% oxygen, is probably one of the most detrimental things that the pre-implantation embryo in vitro could ever see. And I've been working on the embryo for 40 years now, and now we know for a fact that it affects gene expression, histone remodeling, the proteome metabolism, cleavage, and transfer outcomes. So it's really not consistent with good IVF. Now, this review from 8 years ago shows that 3/4 of the world's IVF was done in the presence of 20% oxygen. Now, over the last 8 years, I'm sure many clinics have moved away from 20% oxygen, but we can't avoid it in the lab. We just simply can't avoid it. And that's because we see it during gamete collection, the preparation of eggs and sperm, during ICSI, FERT checks, grading, biopsy. And still some labs, I hate to say, do use 20% oxygen. And if you're one of those labs, please reconsider that. But we also have the generation of reactive oxygen species. We do that during the preparation of sperm itself. During cryopreservation, that actually induces reactive oxygen, and the metabolism of the embryo itself causes problems. It generates reactive oxygen species. And that can have negative impacts on our embryo quality. It produces fragmentation, damage to the membrane's DNA, and as you can see, there's a whole list there. So it's really something we need to avoid. So, enter antioxidants. Now, this is not a new concept. As you can see, the first paper on this topic was in 1991 by Noda, and he was looking at superoxide dismutase, trying to get mouse embryos through the 2-cell block. And it was, in fact, that paper that caught my interest, and in 1992, we started on our journey looking at antioxidants. We know that individual antioxidants can have beneficial effects, but what we wanted to do is what happens when you add them as a group. So I'm going to show you a series of papers, and I want to acknowledge my colleague for the last 15 years, Mai Trung, who's really been the driving force behind all of these studies. So antioxidants in art. Well, we picked 3. Acetylcarnitine. Carnitine because it's the most abundant antioxidant in seminal fluid, but it also has protective properties for the embryo. Alpha-lipoic acid, otherwise known as the antioxidant of all antioxidants. It has the capacity not only to be an antioxidant, but once another antioxidant's been reduced, it can, oxidized, it can reduce it back. So it keeps other antioxidants viable. And acetylcysteine, not only is it in itself a very powerful antioxidant, but it's also the precursor to glutathione. And that's one of the cell's major antioxidant pathways. So we looked at those individually and they were all beneficial. So what we wanted to do was, what happens when we put them together? And what we found when we looked at pronucleate embryo culture, that whether embryos were grown in a group or individually, in other words, conventional lab systems versus that we would use in a time lapse, that when we used 20% oxygen, there was a significant benefit on blastocyst cell numbers when the antioxidants were present. We then repeated that at 5% oxygen and got exactly the same data. So what's interesting is that even at 5% oxygen, the antioxidants are having a profound effect. Okay. Right, we then do embryo transfers, and as you can see, when you look at the control compared to the A3, the AOX group, you can see there was an increase in fetal weight. Now that is not an overweight like we worry about in human IVF, because actually still below the weight of an in vivo developed fetus. But what we are seeing is we're getting closer to in vivo. So we saw a 14% increase in fetal weight, we saw an increase in placental weight and crown rump length, and we saw a difference in implantation. So following that, we wanted to know what happens during IVF. So we went back using a mouse model, but this time did IVF. So in this case, we could do gamete collection with or without antioxidants. And what we showed, as you can see here, is that the control group in IVF had no antioxidants. And when we added antioxidants to just the IVF, we get a significant benefit. If we add the antioxidants just to the culture period, we get a significant benefit. But if you add antioxidants during IVF and embryo culture, you get the greatest benefit. So it's a bit like the amino acid story. You need them to be present throughout all of the procedures. We then went on to look at vitrification. because we were aware that cryopreservation induces a spike of reactive oxygen species. The driving force of this is as we're moving more and more to single embryo transfer, we're cryopreserving more blastocysts. So this is increasing. We're also through biopsy and PGT and a growing demand for oocyte freezing as well as sperm freezing. There are concerns that cryo induces downstream effects. So we wanted to investigate what antioxidants could do. And as you can see from this, again, what we did is we grew embryos in the mouse model to the blastocyst on day 4, then vitrified them, warmed them, and then grew them out to day 5 and analyzed their cells or did embryo transfers. And what we can see is that by the inclusion of the vitrification of the antioxidants in the vitrification solutions alone, we get a significant benefit. When they're present in the vitrification and warming solutions, we get a benefit. But when the antioxidants are present in the warming solution alone, you don't get a benefit. So that indicates that the real benefit of the antioxidants is during the vitrification process itself, which is what we would have predicted. Interestingly, we know that cryopreservation has an adverse effect on the epigenetic state. And in this case, we used histone acetylation of H3K27 and H3K9. And what we saw was that when we vitrified our mouse blastocysts, we saw a significant decrease in acetylation levels. However, when we included the antioxidants, we got an increase compared to the treatment group. Now, it was still less than the control, but at least we saw some amelioration of the effect. And again, we went on to do embryo transfers and showed that we saw an increase in fetal weights once more and crown rump length. So there was clearly a post-transfer effect by including the antioxidants during vitrification. And that occurred both at 5 and 20% oxygen. So where do we go from here? Well, what we wanted to do is actually then follow on and look at the downstream consequences of culture and vitrification with or without antioxidants. And this was really an epic study that Mai did. We recently published this at the start of the year in RBM Online. And we used RNA-seq to actually characterize gene expression in the fetuses and placentas to see any downstream programming effects. And what was shown was that here on the left, in the liver, there was downregulation of over 1,000 genes by cryopreservation alone and an upregulation of 900. And into the placenta, there was a few, 80 were downregulated and 131 were upregulated. But when we looked at the livers and placentas of fetuses that were derived from embryos that were cryopreserved in the presence of antioxidants, so that's the only difference. We cryopreserved them. So this is the whole thing, this one brief treatment. And we had this profound effect in the liver of the fetuses. But nothing really happened in the placentas. But what we can say from all of that is that vitrification, when you include the antioxidants, you can normalize to a certain degree the differentially expressed genes within the liver. So that's really very, very encouraging. And it turns out that these genes that are affected, several of them are involved in preeclampsia. So it has some clinical relevance. We've also done some clinical studies, pilot studies. This was a sibling oocyte study we did with colleagues in Japan, where only the oocytes saw the antioxidants. And then they were cultured in G1, G2 with antioxidants, so we didn't treat the sperm. And what we found when we did the embryo transfers, the control group is G1, G2, is that you can see the ongoing pregnancy rate per embryo transfer was 50% in patients under 35 and about 25% in patients 35 to 40. But when we looked at the data for those eggs and embryos that had seen antioxidants, we saw a similar pregnancy rate in the under 35s, but we didn't see the loss of pregnancies in the over 35s. And this has been followed up by Mitsumoto, who was one of the authors on the paper, but this time looking at both sperm and oocytes being exposed. So rather than just the oocytes, he actually treated the sperm with the antioxidants as well. And this was data presented at the ESHRE, and you can see there was a slight difference in fertilization rates, almost reaching significance, but there was a difference in embryo quality and a trend, again, to higher pregnancy rates in the older age groups. So again, it seems that the antioxidants are having a beneficial effect in the older age groups. Now, we've just completed a very large prospective randomized trial at Melbourne IVF, and we'll be able to share those data with you shortly. So in summary, in all the mouse studies, we've shown that this combination of S-acetylcarnitine, alpha-lipoic acid, and N-acetylcysteine together had a beneficial effect both in IVF and embryo culture, and improved embryo development not just at 20% oxygen, but at 5% as well, which really shows you that that oxidative stress is present throughout. The effects were best and more consistent when you have the antioxidants throughout the procedure, IVF and the culture itself. I didn't have time to show you the data, but actually we showed that the antioxidants led to an increase in intracellular glutathione and a decrease in intracellular hydrogen peroxide, which was really important. As a result of all of this, fetal development, both at 5 and 20%, was significantly improved after transfer in the presence of these antioxidants. We showed that antioxidants helped the embryo survive and go through vitrification, and it preserved their gene expression downstream in the fetus's liver. And in the human studies, the inclusion of antioxidants is associated with improved outcomes in patients of advanced maternal age. So it really is a very interesting and exciting time in this age of antioxidants. So thank you very much.